Okay, it is my great pleasure to introduce our last speaker of the symposium, Dr. Rebecca Dikow from Yale University. Dr. Dikow is the Director of Computational Methods and Data at the Yale Library. She has a BS from Cornell University and a PhD from the University of Chicago. Many of us already know Rebecca because she previously led the data science lab in the Office of Chief Information Officer, or OCIO, at the Smithsonian Institution. She was instrumental in facilitating collaborative research on digitized museum collections and big data using genomics and machine learning tools. She contributed greatly to training many users in data science, bioinformatics, and high-performance computing. Today, Dr. Dikow will speak on connecting dots, people, data, and natural history collections. Rebecca.
Thank you, June, and thank you so much to the organizers for inviting me and to the previous speakers for really setting up this talk beautifully, and to you all for being here. I know I'm last, so I really appreciate you hanging in there. I'm going to really front load my talk with the conclusions and then tell you a story about data and sort of how I got there. You've heard this already today, but it can't be said too often that digitized collections can really help us tell the story of science and the people who did it. For many of the women who collected the specimens for Natural History Museum, the specimen labels may be the only record we have of their work, particularly if they were not directly employed by the museum, and we'll see this evidence today. There are many ways to assess scientific impact, but particularly for women, historical women in science, counting papers and awards is really kind of, will sometimes miss their contributions. So I really think this work is important to really capture the support science network and how important that is to how science has developed. And none of this would be tractable without digitization. So that has really provided such an entry point into being able to make these historical connections. And I'll talk today a bit about how the data need to be improved in order to do that better. But I say that with just huge appreciation for everyone who has worked toward digitizing, imaging, transcribing specimen labels. And really, we wouldn't be able to even think about improving it if we didn't have that first step. So just want to start out with that. Also thinking about how with AI tools becoming so ubiquitous, people are really hungry for data sets to consume. But really, the quality of the data is just super important to how those data can be used successfully. And I really think that the expertise of museum staff is really just absolutely crucial to describing the data and making sure that we, when we provide these data to end users, to data scientists, to whoever is using them, that it is just as clean as possible, as accurate as possible. So that work is really, the people work is just essential to any sort of computational work that comes later. I also think that as we're in this very uncertain time for science, really focusing on the community aspects is really energizing. So I hope that that's something you take away from this talk. So I'm going to be talking today as an entry point about Mary Vaux Walcott. She was a botanist, an artist, and a philanthropist who was around the Smithsonian for quite a long time. Her husband, oh, sorry, this is a specimen that she collected, as well as a painting she did based on that specimen. So really lucky to have both of these things digitized, that we can really kind of find her natural history work and connect it to her artistic work. And it's just such amazing resources about Mary at the Smithsonian. Her husband was Charles Doolittle Walcott, who was a paleontologist who's very well known for discovering the Burgess Shale and was also Smithsonian secretary. So while Mary Walcott was not employed by the Smithsonian, she was very well known and really a big part of it while her husband was here as well. Also, as an entry point to this work, I'd like to talk about Vicky Funk and Effie Capsalis, and it's really an honor to be able to talk about them together in the same project. Both of them left us far too soon. I met Vicky when I was a brand new postdoc here at the Smithsonian. She sent me a cold email and said, I heard that you're around and you know something about data. And we met, and I got involved in a project that led to 4 or 5 papers. And she was extremely collaborative, and that's the same is true of Effie. Effie worked at the Smithsonian Archives and then in the Provost's office and led the Open Access Initiative at the Smithsonian. Together, they worked in the early days of the American Women's History Initiative, which has become the Smithsonian American Women's History Museum, which is in development. They worked on a curatorial committee to try to understand, well, one of the goals was to try to understand women's impact on Smithsonian collections. And when the first day I met Effie, she again heard I knew something about data and said, how can we kind of quantify how many collections were either collected by women or created by women for some of the other museums? And I was like, well, let's see what we can do. And so that's sort of how I got to know both of them. And I'm really just happy to be able to talk about their influence on this work. So the Funk list is a list of Smithsonian women in science through Smithsonian's history. And this is something that Vicky and Effie worked on together to create. It was named the Funk list after Vicky passed away. This is a list of, you might say, well, wouldn't we know that already based on employment records? But that's sort of the key piece, is that women often weren't employed, particularly for the first 100 years of the Smithsonian. Their husband was employed, sometimes their brother was employed, or they were volunteers. And so really wanted to find a way to make sure we're capturing as many of the women as we could. And so then I started working on projects to take that list, find the specimens collected by the women on that list, and see what we could do. So here are 2 specimens which were collected on the same collecting trip by Mary Vox Walcott. But you will see, we zoom into the labels. Yeah, it's pretty legible. On the left, it's written Mary Vox Walcott, July 1937. And on the right, it says Mrs. Charles D. Walcott, 1937. Okay, both of those are accurate ways of depicting her name. But at the time, and this has been corrected, but at the time, in the specimen database, the one on the left said, I think MV Walcott, because it often abbreviated. And the one on the right, it said it was collected by CD Walcott. The only reason we found this was we had an intern at the time who was working through this Funk list and noticed that Charles Walcott passed away, I think 10 years before this collection event. So that was like a light bulb went off, right? Like, what's going on? Now, it could have been that it was, the date was wrong in the database. It could have been many things, but the amazing thing about how these have been digitized is we also have the image, so we can go back to it and say, kind of cross-check. And so we're not the first people to find this kind of, this happening, but it really sort of set off a lot of work to sort of think about, okay, how common is this? Is this true for other people? And I'll show you more of that work now. Here's another example, which I think is really just illustrates this challenge. So Mary Vox Walcott and Mrs. Herbert Hoover, the First Lady, were friendly, and they went on collecting trips together. I think pretty locally, but this specimen, you can see, has Mrs. C.D. Walcott and Mrs. Herbert Hoover. And initially, again, this has been corrected, but in the collection system, it was as if it had been collected by their husbands. And so I think why this story, I think, resonates with me and with others to whom I've shared it is really, these are very well-known women. So Mary Vox Walcott was independently wealthy, and she really did so much for the Smithsonian. She was well-known. But even in those cases, we're sort of missing information about their contributions. So what about the women who aren't so well-known? And so it really just, to me, drives home the importance of this work. These are some of Mary Vox Walcott's paintings. There's many of them. I encourage you to check them out on collections.si.edu. Of the 470 specimens in the the National Herbarium collection, 470 she collected, 59 were originally attributed to her husband, and that's since been rectified, which is great. I'd like to just talk through some other examples. So Anna Murray Vail and Elizabeth G. Britton. Anna Murray Vail was the first librarian at the New York Botanical Garden, and Elizabeth Britton was important in its founding. Her husband, Nathaniel Britton, was one of the co-founders. And I just wanted to show two labels. Oh, and I will share that Elizabeth Britton has more than 22,000 specimens that she's collected. And she wasn't employed by the garden. She resigned her position when she got married. But just, I think another important point to drive home is really the dedication of many of these women who were not employed, but to continue to do the science that they were doing. Here are two labels, which I really liked because sort of this other through line, I think, is really these connections between pairs of women working together. And the digitization allows us to really find those connections in interesting ways. This label on the left, you can see is written Mrs. Britton and Miss Vail. And on the right, this is just so wonderful with these typewritten labels. We're they didn't know about digitization and transcription and how hard handwriting would be, but this is just amazing forethought. So it's nice that they have this, wonderful label that was pre-printed before their trip. This is also a very common thing that's quite difficult, is there are a lot of Mrs. and Miss records without first names. These are tough. So you'll see this one on the left from the Yale Herbarium with Mrs. Hobbs and Mrs. Black. Also the ditto marks, not great for digitization. That's okay, they didn't know. And on the right, you'll see this one, Mrs. Dr. Peck, which is also sort of fascinating. And it's hard to find out much more about these women in these situations. There's some detective work that can be done, but it can be quite challenging. Here's another example where you'll see on the printed label, it says collected by Mr. and Mrs. H. Reese, I think is the pronunciation. But in the database, it says Mr. Reese and H. Reese. And so oh, okay. The wife's name was Millie Timmerman. She also often went by Mrs. H. Reese. But you'll see this is just sort of this challenging thing with transcription that things can get disassociated. So how can we potentially try to tackle this? And I will talk to you about this amazing platform, which if you haven't heard about already, I'm excited to tell you about, called Bionomia. This is a website you can go to, sign up, and help attribute specimens to people. This is Vicky's page. You can see it gives a really nice snapshot of her collecting career. Bionomia pulls data from GBIF, so it's not just Vicky's contributions to this herbarium, but it's her contributions all over the world. So she took trips to identify specimens at other herbaria. Those identifications are present in this platform. The really exciting thing about it is that it provides this way to look at aggregated data from different museums and reconcile those differences in how people's names are written. So, and this little yellow arrow is showing you this Q number, which is a unique identifier for Vicky. So some of you may have Q numbers and you don't even know it. Check it out, or if you're in Wikidata. So this is a way of, we know that, for Vicky's name, it's not as big a problem as it is for other people, but you see there's multiple ways that her name may be present in different collections. Even within the National Herbarium, there's just differences in the way things have been tracked over time. This Q number allows you to associate all of those ways of writing a name into a unique identifier. Super important and critical as we, work across different collections. So we worked on a paper which was published in 2023, where we took all of the women on this Funk list, which the Funk list now has, I think, more than 600 women in Smithsonian science on it. We looked at only those who were deceased for this work. And we went into bionomia and attributed specimens to them, both specimens they collected and identified. For 40 women on the list, we were able to do this, and we attributed more than 120,000 specimens. And you can see here two different plots. One that's just sort of with all of the number of specimens per woman on the list. And the one on the right breaks it down by department. You can see botany is really up there, and that's first because it's fully digitized. But this is only possible based on the digitization. So other departments are further behind, and for real reasons of digitization being more complicated for different kinds of specimens. But also there were more women botanists employed at the Smithsonian before, also invertebrate zoologists before other departments. So that's sort of also reflected here. Another challenge with sort of the different departments is, particularly for entomology, there are some issues with just the way that the way the work progresses, right? It's not a failure in any in terms of digitization or anything, but often specimens are collected in series and they may not all have individual labels. So you're sort of potentially missing some numbers there. And for type also the labels are much smaller, right? So the botany specimens, I think, first, the two-dimensional aspect, great for digitization. Also, the fact that there's space to write more on the labels is great. So there's sort of discipline-specific differences that are also cause some of the patterns here. So as we went through that work, it was sort of in some ways disappointing that we could only attribute specimens to 40 different women, even though we were looking at more than 100 initially. Again, part of it's a lack of digitization in certain areas, but we did want to sort of understand how are these distributed across the tree of life? Like, what do we see? And you can see it's amazing. It's across the whole tree of life. So this is just for these 40 women. And I think, this on the second side, you can see the count of families of specimens collected. And, Vicky's right at the top with more than 250 different families that she collected in her career, which is just, it's remarkable. We also plotted sort of a geographic distribution of the specimen attributions built into world regions. And these are, our top 4 women in botany. And you can see differences in terms of numbers of collections versus identifications, and particularly for the earlier women, potentially traveling was not as common or possible for them. Mary Agnes Chase, there's a story about she wanted to go to BCI, a part of the Smithsonian Tropical Research Institute, to collect, and women weren't allowed to stay overnight there, so she had to send someone on her behalf. So there's just like so many interesting stories you can also learn from this work. I wanted to highlight Annie Lorenz, who was a botanist in the Connecticut area who collected more than 3,000 specimens. But I really couldn't find a record of her working anywhere. But through resources like the Biodiversity Heritage Library, we're able to find both this biographical notice of after she passed away, which has appreciations from other botanists. So it's as just kind of a volunteer naturalist and collector. She was really important in the biology community. And through the Biodiversity Heritage Library, it's a little bit hard to read, but I found this series of letters between Annie and James Franklin Collins, who was a professor. And also, she has sent some specimens, and in the right, you can see I had sent it to Mrs. Britton, but she says that she's too busy to look up anything and referred me to you. It doesn't seem like Annie Lorenz ever met Elizabeth Britton, who I mentioned much earlier. But there was just this network of collaborators, and it just sort of really interesting to find this record of it that we wouldn't have found any other way. I also really loved the Collins' letter back to Annie Lorenz, which was, "Thank you for sending these. I wish I had time, but all these undergraduates are, making my life difficult." But it just sort of highlights to me just how, because we have these archives and letters digitized, we can really understand more about the lives of these people and how the science progressed, not just the sort of specimens. I did want to call out Lux, which is a really exciting cross-collections discovery platform at Yale that links library and museum data in a knowledge graph. So this is sort of getting us to that place where you can search for a person, and if there are specimens they collected and also there are books that they wrote, you can kind of get to that all in one place. But I will sort of emphasize again that this is only possible with good data going in. So good metadata, that's people have worked hard to ensure it's really linked to that person. So the takeaways from this talk is really just emphasizing that GBIF, Bionomia, Biodiversity Heritage Library, to me, they're essential infrastructure to enable the future of collections-based research. And that AI and other computational tools depend on good data, which really depends on a lot of people time. And I encourage you to meet or to learn about someone new today, and please go onto Bionomia and attribute specimens to them. I find it so fun and relaxing. I know I'm like a nerd, but I think you'll enjoy it. And with that, I'd like to thank the co-authors on the 2023 paper, Darren Berber, the former Smithsonian CIO, who supported a lot of this work, and previous funding from the Women's History Museum. And while I take questions, I will just put up some places we have found Mary Walcott referenced in the Smithsonian annual reports. And it's just, she brought the first elephants to the Smithsonian. She bought a boat. She just like figured things out. And I just love reading about her. So thank you for your time today.
We have time for questions.
And this is probably the tip of the iceberg, because there are many examples. One which is the Spanish-speaking people usually use two last names, and usually we are cataloged under the second last name, but not for the Portuguese people or Brazilians. They are the last one. So it's a lot of variation in the theme. So trying to track down the history of collections of a collector where they worked is very difficult. So there are a lot of other examples of things that could be done to improve the data. So I work with climbing plants, and it's very hard to find in the data if that is a climbing plant or not. It's probably the label says something, but there is not a field that talks about the habitat. And this is more increasingly very important when people are working with trees or shrubs. You want to have a field where you can find that information and filter those out. And so there are many things that are, but this is very exciting, seeing how you're doing with all this data and how you navigate it.
I agree. There's a lot to be done, but I think we have a great starting point now. So there's definitely computational tools you can use to improve metadata, but I can't totally get around having to really dig into solve a few mysteries first. But yeah.
It's a matter of sitting down and studying the whole thing, and it's going to take time. Well, thank you for the talk.
Yeah, there's questions.
This is a really fascinating talk. I just thought of somebody I was aware of for my own work through who should be in the list and the database if she's not already. But my question has to do with like the use of automated tools for processing these kinds of data. And so a lot of us have heard a lot about biases that emerge in these kinds of large language models and so forth that actually replicate the biases of the human societies that generate them and the human individuals that generate them. And I was wondering, do you know anything about whether such tools are useful for recognizing unrecognized women scientists, or if they, like some humans do, would misattribute records to male scientists if you tried to like use them on these data?
Sure, I think there's certainly going to be biases present in any method. But I think as long as there's a person, human in the loop is, the phrase that comes up a lot, to really cross-check things and to, but I do think, yeah, there's not a way to take that out of it. It really does require some detective work, and I don't think AI tools are good at that kind of detective work. But the more things that are digitized, the more we can sort of cross-check it, maybe automatically. Yeah.
Thank you.
Thank you.
Thank you, Rebecca.